Assalamu alaikum and welcome. Here we come about with another type of the topic and other type of practice of bioenergetics. First I will start with first MCQs. Here is our first MCQs. The most abundant protein in the chloroplast is it is mentioned in the PDB that the Calvin cycle that most abundant protein in the chloroplast and probably on Earth is the rubisco. It is the ribulose bisphosphate carboxylase, or it can be ribulose bisphosphate oxygenase. It has is an enzymes that catalyze the reactions of ribulose bisphosphates with carbon dioxides during the Calvin cycles. So it is best answer is rubisco is the most abundant protein on Earth, and here is its answer first. Okay. Then we came across the product of the photosynthesis in green plants are the product of photosynthesis. You know that photosynthesis produce C6H12O6 and O2, 6 molecule of O2 and H2O, and also energy. Here is the exact product of photosynthesis in green plants. But if we summarize the reactions, then we came across only this type of reactions. Whether it is confirming about this type of product or this type of product, but actually because we use 12H2O, so our D options will be correct without summarizing the equations. Now check it whether it is A or whether it is D. So it is the D options because we have to use 12H2O, out of which 6H2O are our products. Then our third MCQ is incorrect statement for the photosynthesis is we have to look forward this statement that is incorrect for photosynthesis. It occurred during daytime, it is correct statement. It requires chlorophyll, it is also correct statement. It uses water and carbon dioxide, it is also correct. It uses oxygen, no, it do not use oxygen, it produce oxygen instead. So it is our correct option is D, and here is the third correct option is D. So we are best with 3 MCQs. Then role of H2O. Spectrophotometer is used to measure absorption of carbon dioxide, no reflection of the pigments, absorption of carbon dioxide, absorption of different wavelength. Actually, the spectrophotometer is instrument that measures the absorption of different wavelength of the light by different photosynthetic pigment. Here is our option: absorption of different wavelengths by different photosynthetic pigments. So here option correct option is D, and here is also a correct option is B. B. Here is correct option is B. What is the B? Absorption of photo. Spectrophotometer is used to measure. No. It is used to absorptions of different wavelength of light by different photosynthetic pigments. Its correct answer must be of D type. We have to follow the explanations. Spectrophotometers is an electrical instrument. What type of instrument? An electrical instrument which is used to measure the relative ability of the different pigment to observe different wavelength of light. Yes, here is the wavelength of light, and correct option is D. Our correct option is D instead of B. So we have to correct this. But anyway, so we have to correct this. It is D. Fourth option is D. Then we came forward another MCQs which shows the effectiveness of the wavelength of light driving effectiveness absorption action spectrophotometer. If action spectrum give off the relative strength or effectiveness, and absorption give out us the knowledge about how different wavelength absorbed by a different photosynthetic pigment. So the correct option is action spectrum here. Correct option is action spectrum. Fifth is B options. Oh, here is also exchanged with the fourth option is B, D, and fifth option is B. But here is exchange. Fifth option is D, not a B, not a D. We have to follow the explanation. Spectrophotometer is an electric instrument graph showing the relative effect of different wavelengths of light driving the photosynthesis called action spectrums. You know, action spectrum is the correct option. It is of about D. Here is the wrong key. And then other MCQs that are air contain percentage of carbon dioxide, we have followed it before. That contains 0.03 to 0.04% of carbon dioxide in the air. Then check whether it is correct key or not correct. 11th is of D, that is correct. So which of the following groups does not use water as the proton donor? Water as a proton donor are not used by cyanobacteria plants, purple sulfur plants, and algae. Algae use purple sulfur bacteria actually not use water directly. So we have to tick purple sulfur bacteria. When Neil hypothesis, they were working on bacteria and noticed that they do not release oxygen because they do not use water. So the correct option is here is D. Where is it D? I mean algae? No. They use water. We have to follow the photosynthetic bacteria use hydrogen sulfide as a proton donor, not a proton donor. So we have to take the bacteria, not algae. So the correct option is of bacteria, and correct option is of B. Correct option is of bacteria, meanings purple sulfur bacteria, meaning B, not a C. Here is the correct options. Purple sulfur bacteria. Carotenoid absorb mostly blue violet live BV lights. They are red to orange, but they absorb strongly blue violet color of the radiation. So the option is correct. Here it is correct. Then came across here. The following is the correct sequence of the energy transferring between the photosynthetic pigments. We know that first of all carotenoid absorb, then chlorophyll B and chlorophyll A. Here is the B option is very correct for 15th. 15 is B option. Yes, it is all right. Then we came across the location of the phytol tail of chlorophyll B is. Where is the phytol tail of the chlorophyll B? Is embedded in stroma or thylakoid surface, thylakoid membrane, thylakoid lumen. I think thylakoid membrane is it is embedded in the thylakoid membrane. Phytol tail is embedded in the thylakoid membrane. We have to follow the key. 17 option B. It is all right. In the thylakoid membrane. Then location of photosynthetic pigment in the purple sulfur bacteria. Where the photosynthetic pigment are found in purple sulfur bacteria. Grana, there are no grana cytoplasm. So because there here the photosynthetic organelle, which is chloroplast, is absent. So grana, thylakoid, and stroma are also absent. So the cytoplasm is the process or the location of the photosynthetic pigment and purple sulfur bacteria. It is the basic concept we have to follow. 19 option is D. All right. Then we come to another MCQ, that is 22. During the cyclic photophosphorylation, electrons passes from all except Here we have to follow the cyclic photophosphorylation, not cyclic. They come across photosynthesis system 1, yes. Photosystem 2, no, they do not pass through the photosystem 2. Paradoxin, yes. And cytochrome complex, yes. So the correct option is photosystem 2, where the electron does not passes. 22, correct option is C. Here is 22 and correct option is C and best. Then we came across, where do the light reaction take place? We know that light reaction takes place at thalloid membrane, and dark reaction takes place at stroma. Like we can also say that light reaction takes place as garena, and dark reaction takes place at stroma. So the correct option is of B. Here B is the best answer for this questions. 24, B best. B for best. Then we came across, each photon of the light excite how many electrons? Each photon of light almost excite only one electrons. That is mentioned in non-phosphorylation, non-cyclic photophosphorylations. 26 portion is 26A. Yes, all right. Then we came forward to 28, and the NADP reductase transfers the electron from NADP, actually transfer electron NADP to ferredoxins. No, ferredoxins NADP, ferredoxins to NABTP, and it is converted into NADPH. So the correct option is of the B. B option is correct according to this. According to non-cyclic phosphorylation, we have to follow the key 28 MCQs, and it is of B option. B for best option. B for best, and our option is best. And which statement is not true about the non-cyclic electron flow? Non-cyclic electron flow is not correct. It absorbs photons into the photosystem 1. Okay, it produces ATP. Well done. It is a long pathway. Say carbon dioxide fixation. No. Here carbon dioxide is not fixed. Carbon dioxide is fixed in the dark reaction Calvin cycle, actually. So the best option is D. 30. Best option is D. Worse. Wow, great. It is correct. Then we have follow 32. Z scheme is another name for non-cyclic phosphorylations. We know it very well. Z scheme. C option is the best option for this. And then we have to follow the answer. 32, C option. C for cute. Which is not true for the dark reactions. Which is not true for dark reaction. Does not require light directly. Okay, it is best. Also called Z scheme. No. Dark reaction is not called Z scheme. Non-cyclic photophosphorylation is called Z scheme. It is our best answer. It uses APT. Yes. G3 is produced. Yes. So 33 best option is about C. We have to follow 33 best option C, and all good. Then 34, the process in which the carbon from CO2 is incorporated in organic molecules is called Calvin cycle. Yes, it is the process where the carbon dioxide is fixed in organic molecules. So the correct answer is B. 34. Correct answer is B. Okay, best. Then we came across this type of MCQs, like the product of the dark reactions, where the product of dark reaction is G3P. It is a good answer. 36C. Yes, 36C. Good. Then 37. If 12 NADPH are used in Calvin cycles, then how many glucose molecules will be formed? We know that 6 NADPH used and 9 ATP are used, and it's 12 NADPH is used and 18 ATPs should be used. So a glucose molecule will, and G3P is produced by, and it is of 3 carbon atoms, and 6 NADPH are used for 3 carbon, and glucose is a 6 carbon, and 12 NADPH is used for only production of 6 carbon molecule. So 37, best option is 1, because glucose contains 6 carbon molecule, and for production of the 6 carbon molecule, the number of NADPH that are used are 12, and for 12 NADPH we can produce only 1 glucose molecule. So the best option is of 37. NADH is oxidized during which process? Right, at during lactic acid fermentations, it is again converted. NADH is converted into NAD. So during lactic acid fermentations, it is got oxidized. So correct option is of C. 40. Correct option is of C. Good. Bread. Then the final fate of the glucose in the yeast when it is deprived of oxygen. When there is no oxygen, the final fate of glucose is glucose in the yeast. Yeast is alcohol. The final most product is in the alcoholic fermentation is alcohol, because alcoholic fermentation occur in the yeast. Option is D. D rest 4 options. 43. D. Good. Number of ATP molecules 45. Number of ATP molecules used during the preparatory phase. ATP molecules used is preparatory because we know it is all clear that 2 molecules are used for preparatory phase of molecules. It is very easy MCQs, 45. A. All best. Then water is released during conversion. Water is which process? Water is used during 2-phosphoglycerate is converted into phosphoenolpyruvate. It is the process where oxygen is produced. The correct option is of A. 47. Correct option is of A. Good. Then we came across this type of the gross production of ATP molecule during the glycolysis. We have to follow the gross ATP. Gross ATP means total amount of the ATP produced. 4 ATP are produced directly, and 4 ATP is produced directly, and it is the product of the glycolysis. Other are produced indirectly, that are through NADH. So the best option is B. Single best options. Then the oxidation of the succinate produced. When succinates like oxaloacetate, citrate, isocystrate, alpha-ketoglutarate, succinate, fumarate, malate, and succinate is oxidized, and it is produced FADH2 instead of NADH. So it is the most important MCQs also. 51. B. Good. FADH2 is the best answers. Again, precursor of the fumarate during the Krebs cycle is required for fumarate synthesis. FADH2 oxidases, because non-reductases, it's non-reductases, fumarate dehydrogenases. Hydrogen is removed from fumarate. Succinate dehydrogenate, because succinate dehydrogenate and fumarate are produced. So best option is of the C. During this process, the succinate and hydrogen is donated to FADH2 and it is converted into FADH2. The best option is of C. 54. The best option is C. Correct. Then we came across which of the following process is used in the conversion of the pyruvate to acetyl coenzyme A. There are the decarboxylations and dehydrogenations. Decarboxylations and dehydrogenations, the both process A and C. So the correct option is D. A. C. D option is correct. 57. D option, all best. Then before entering the Krebs cycle, the pyruvate decarboxylated into acetic acid or acetaldehyde. Acetic acid, it is actually converted into acetic acid. Acetyl coenzyme 60 is the best answer. D. Okay, all best. Then we came across class 62. Which of the following is the final acceptor of electron in respiratory chain? We all know that oxygens. And 61. During the respiratory chain of the cellular respiration, cytochrome C oxidizes. Cytochrome C oxidizes. It accepts electron from B. Here is the option for cytochrome B, C, and A complex. It takes electron from B, so it oxidizes cytochrome B, and the correct option of 61 is B, and 62 is C, B, C, 61, 62, B, C, 61, 62, B, C. It is very good, B, C, B, C. Which of the following type of the mammalian cells does not carry out oxidative phosphorylation? Which of the following type of mammalian cells does not carry out oxidative phosphorylation? Which type of neuron? Neurons have mitochondria. Yes, it produce. Erythrocytes do not have mitochondria and do not produce the oxidative phosphorylations. Axanthic cell produce. Cardiac muscles and neurons produce. Erythrocytes are the best option for this. 62 A option and very good options. Then ATP formation through the oxidative phosphorylations involve direct reactions. No, light reactions. Yes. Chemiosmosis also as a best term for it produced by chemiosmosis mechanisms of oxidative phosphorylations. So correct answer is 66 C best. Good. Then we came across high concentration of NDH inhibits pyruvate decarboxylase. Phosphofructokinase pyruvate decarboxylase is inhibited by FDH. It is the at last diagram, which is it is very important regulation of cellular respiration. The 68A option is the best. 68 option best. Then we came across the phosphofructokinase is inhibited by ATP. High concentration of ATP inhibited phosphofructokinase. It is very important. MCQs again. 67C. Then we came across aldo sugar, that is intermediate between the respiration and photosynthesis, is glucose. No, glucose, glyceraldehyde 3-phosphate. Aldo sugar, aldehyde, dicarbox, acetone, fructose. Aldehyde group, it is the glyceraldehyde 3-phosphate. Actually, dihydroacetone contain is intermediate, but it is ketonic. So the aldo group is glyceraldehyde 3-phosphate 69C. Okay, best. Then 70 G3P is converted into glucose phosphates in glyceraldehyde 3. We have glyceraldehyde 3-phosphate converted to glucose phosphate. It is occur in a Calvin cycle, and Calvin cycle occurred in chloroplast. The best answer is B. 70B. Good, all good. Then last deamination of the amino acid take place since deaminations. Amino group is removed. Liver, it is the process of liver. 62A vast answers. Liver is the powerhouse and storehouse and of metabolism. Store